So I work on trying to model C. elegans neural dynamics. So C. elegans are these small worms. They're about 1 millimeter long, and they only have 302 neurons. And with that small set of neurons, they're able to produce a pretty wide variety of behaviors. So these are C. elegans in a petri dish. They're swimming around, and they make different behavioral decisions without experiencing any stimuli. So this is forward crawling. This is a segment of them reversing. So this worm kind of stops, it does an omega turn, and it decides like what direction it wants to go in next. So they're making all these decisions without any input, but also they obviously respond to strong stimulus. So here's an example of the C. elegans gets touched on the head and it quickly reverses to like escape from what maybe could be a predator. So those are some basic behaviors. They can also swim. So if the agar they're in is thick enough, then they'll crawl. But if it's if they're like in water, they'll have the swimming behavior. So they can really modulate their muscular activity according to like the medium that they're in. And then here's sped up video of a C. elegans foraging for food. So it's looking for E. coli, and you can see the different paths it's making in the dish, and it has some sort of search behavior that it's doing to explore the plate. They also can form social aggregates, so they like to clump together, and there's various ideas about why they form these social aggregates. So a basic behavioral sequence that they exhibit is they will go forward, at some point they'll stop, they'll do a little reversal and then a turn and then go in a different direction. And you can track where they go on a plate, look at the time series of these behaviors over time, and do simulations of about the regions that the C. elegans explores in space. So something that we want to understand is how is the neural activity like making these decisions and supporting this behavior. So here is the C. elegans connectome. It's fully mapped. All of the neurons have names. They're more or less connected in the same way across worms. And so one would think that this is a simple enough organism with enough similarity across worms to kind of completely understand the activity, how it's produced, and how behavior is generated from it. But this still is a very much an open question, despite all of the information that we have on this worm. So this is a video of this NeuroPal technology where all of the neurons, they can be most of them, they can be labeled at the same time as their activity measured. So this is pretty cool.
Activity is firing rate?
Pardon?
Activity means firing rate? Or...
So here activity is calcium imaging. So something interesting about C. elegans neurons is most of them don't fire. They don't have like the traditional spike and the reset. They have like a graded potential, like a higher activity state and a lower activity state. So it's not all of the, it's not immediately translatable to other organisms because they don't spike. Yeah. And then it's also an open question, why do these neurons not spike? And why do like most neurons in like mammals, why do they spike? So that's still kind of a mystery here. Yeah, so not spiking neurons, they have a graded potential. And then here's this is showing like calcium imaging of like 10 random neurons. The Flavel lab, they have all these whole brain imaging data sets where all of the neurons are measured at the same time. And because the neurons don't spike, the calcium imaging time series are a better reflection of the voltage levels than in a spiking. So that's kind of nice. Something that I find interesting about these data sets is like most of the neurons, they tend to be pretty active with richly varying activity despite not having strong stimulus. So there's our experiments where the worms are given a strong stimulus, they're poked, they're given a heat shock, something is happening and they're responding in a certain way. But the neural activity isn't just quiet without stimulus. The C. elegans, it's moving around, it's making decisions lots of things are happening in its brain, despite whether or not there's this strong stimulus coming in. So that's kind of interesting. How is that happening? What about the structure of the network is supporting that? And because we like have a pretty relatively better understanding of how individual neurons respond to input. We have all these math models, a Hodgkin-Huxley model, leaky integrated fire, so we can understand like when a neuron will or won't fire, and then there's modifications of those for the C. elegans neurons because they don't spike, so the parameters are slightly different. But we can understand individual neurons, but then when it comes to a network and this collective activity, there's still a lot of open questions. Something else that experimentalists have like noticed is that there are many neurons that are correlated or associated with specific behaviors. So it's not just one neuron per behavior. There's a lot of different neurons that are more closely, like tightly or loosely associated with different behaviors. And then also, many neurons are associated with multiple behaviors. So it's like not each neuron doesn't just have one task, it's doing many tasks. So that's interesting, I think, that these tasks are distributed throughout the network. There are many neurons are doing the same thing, and then many neurons are also doing a lot of things, which I think is kind of an open question. So two behaviors that are the neurons and the behavior association are a little bit better understood is forward and reversal locomotion. So experimentalists have identified like this core set of neurons that when they're active, the C. elegans will move forward. And then when they're not, when instead these reversal neurons are active, it'll swim backwards. So in initial modeling, we've tried to make a model for how this switch happens in the worm, but there's a lot more that the worm does other than the forward and reversal switches. There's turns, a lot of decisions are happening. So I think this is a very interesting system to study and try and understand the sensory input, information integration, and then behavioral output. So that's all I got. Thanks for your attention.
So I have a question, which is, I guess presumably people have done these sort of like, let's splice away several of the, or we shut down some of the neurons because we know that there's some redundancy or whatever. Like, I guess, have people done this? Am I right about that? And like, what did they see? If you know.
Yeah, no, that's a good question. Yeah, so there's a lot of ablation studies where they knock out specific neurons, like the AVB neuron, it's hypothesized that drives forward. So they knock it out. And the C. elegans still swims forward, but just not as often or not as long. And so there's a lot of redundancy where they'll knock out specific neurons and like, oh, you get a decrease in this, but it doesn't completely eliminate the behavior often, which indicates the system maybe is robust in some way, that if you knock one thing out, it's not going to completely destroy the worm's behavior.
Thanks, really cool talk. I'm assuming these neurons still have voltage-gated sodium channels, but are they behaving more like in non-excitable cells, like astrocytes or leukemia? Is that known?
So I'm not an experimentalist, but from my understanding is they don't, the structure is different. They don't have sodium channels. It's more through the calcium channels that I don't want to say like across the board they don't, but the like activity, the rise in voltage isn't generated by the sodium channels. Yeah.
I think we have time for one more question. Anybody?
Yes.
I saw you showed us the time series. Similar to the question Maria asked, I'm curious if people have done this. If you have all these time series for the 302 neurons and you build a correlation matrix between each one, and then you decompose it, does this reveal, do you think, modes of variation that correspond to different functions, or the things the activity the worm can do?
Yeah, so people have done this. They've done like SVD or looking at correlations, and they find like different clusters of neurons that are associated. Like for each behavior, they like rank how much each neuron is like correlated or associated with that behavior. So there's like maps of this data and where each neuron shows up on the behaviors. It's like involved in and behaviors the neuron isn't involved in.
I get it.